Okay, I think we'll start. People are still joining, but I think we'll crack on. So, good morning. I'm Penny Cunningham. I'm based here at the Biorenewables Development Centre, and I'm the project manager for the H2Boost Phase 2, which is a Department of Energy Security and Net Zero funded project. The aim or the mission of H2Boost is to produce biohydrogen and to capture carbon. And in this webinar, you'll be hearing from some of our partners. As you can see, the partners are listed below. There are 10 of us in the consortium on how we're actually going to do this. And we'll be doing it at quite a large scale. So I just go through the start. So here's the agenda. We'll hear first from Debs Rathbone, also here at the BDC, who will introduce the H2Boost project and explain what we achieved in phase 1 and where we're going with phase 2. Then Alex Dukes will explain the work that we've been undertaking here at the BDC. James Rundell from Cube Renewables will talk about the actual kit that has been built, the AD and the dark fermenter, and the setting up of all of that. And then we have the carbon capture elements with Miller at the University of Leeds and David Evans from Sinocapture. We'll complete the workshop, the webinar, with Chris Corner and David Turley talking about the LCA and the TEA, and then we're open for questions. So if you do have any questions during this webinar, please put them in the Q&A and we'll endeavour to answer as many as we can at the end. So we will start by me handing over to Debs, who will go through what the H2Boost project is. Thank you.
Okay, good morning, everyone. As Penny says, I work here at the Biorenewables Development Centre. For those of you who don't know the BDC, we're an open access, not-for-profit organisation focused on the development and scale-up of bio-based products and processes. Very happy to tell you more about that at length. Just drop us a line and we can tell you more about what we do. But for today, the next few minutes, I'm going to talk to you, as Penny says, about how we've got to this point. What's the background work that's gone into phase 1 of H2Boost, and what are our aims, our overall plans for this project? And I'll introduce the project partners and their various roles within the project. So phase 1 of H2Boost was led by the University of Leeds, and it was funded by BEIS. And really it was a 6-month feasibility study to see whether we could build a commercial case for the use of available waste feedstocks within England to make bio-based biohydrogen. And you can see on the left-hand side there, the left-hand panel, the sorts of feedstocks that we were evaluating. So things like food waste, manures, slurries, residues from a whole different range of sectors. To get that information, we interrogated a wide range of data sources, including local authority reports, various reports from RAP, DEFRA, useful interactions also with Natural England and the Forestry Commission, as well as individual conversations with SMEs, large companies, and sector bodies as well. Once we had that information together, we overlaid it across the locations and types of AD plants. And the thought at this point was that we would build the biohydrogen element at an existing AD plant. And so this is a useful link if anyone's interested in AD information. So virtualtimeregion.com. It's an interactive map where you can look at different feedstocks and volumes going into all the AD sites across England. By overlaying the feedstock availability and amounts with the various AD sites, we really identified a zone of the east of England, really from Yorkshire down to the southeast, that would be a good place to site the demonstration scale plant when it came to that. So the second part was to prove that we could actually make biohydrogen, and this was going to use a process called dark fermentation, which is essentially a mixed microbial culture an anaerobic culture, which is digesting the various materials. It doesn't require light for this process. Acts very much like anaerobic digestion, except there's no methane produced. One of the main intermediates is hydrogen. And so we were spent a long time looking at the inoculum preparation, how we might take that mixed microbial culture and knock out the methanogens. We looked at various things such as pretreatments of feedstocks, such as enzyme addition. And you can see here, the graph on the right just shows the difference between just mixed food waste with and without enzyme addition, and the typical yields that we were getting in small batch anaerobic fermentations. You can see here that the majority of the activity, or the majority of the hydrogen production, was in the first 2 to 3 days. So that's really good from a residence time point of view within the system. One of the things we're very keen on doing throughout this phase 1 was to try and understand what was going on with the microbiology. And so we undertook a lot of microbial profiling, both in the inoculum preparation stage, but also throughout the various dark fermentations, to try to get a handle on what was happening with the microbiology and how it might be varying across the different runs. Next stage was to take the residue from the dark fermentation, so after the hydrogen had been produced, and to see whether that was a suitable feedstock within anaerobic digestion. So this time trying to make methane. And you can see from the graph here that there is a bit of a dip, there is a bit of a difference between feedstock that just goes straight into anaerobic digestion. That's the solid line. Then the dotted line is material which has already gone through dark fermentation. And that will vary according to the feedstock and how well the hydrogen has been produced. And again, all the wraparound information we could generate from that, not just the gas yields and composition, but also analysis of the digestate from anaerobic digestion and volatile fatty acid compositions, anything we could really determine from the setup to try and guide us on what was happening. So other work packages within phase 1, we're really trying to take that information, look at also at the carbon capture element, but also whether there is commercial feasibility in developing this further into a commercial scale plant. So the final report for H2Boost phase 1 was to come up with a preliminary design and look at the capex and opex around that, as well as things like the techno-economic analysis and life cycle analysis. So on to phase 2. As Penny has said, this is a DESNZ funded £5 million project. Fairly short though, it's only 21 months. And within that time, we'll be looking at dark fermentation at a much larger level. So this time we have 2 6,000 litre vessels, which are being fed with a range of feedstock and inoculum prepared at scale. Some of the feedstock being treated by enzymes and really sort of focusing on the lignocellulosic element of feedstocks to help that digestibility of that material. The hydrogen that will be generated will be cleaned up. Unfortunately, we can't take it as far as fuel cells within the boundaries of this project, but the eventual aim is that the hydrogen will be produced for the transport sector. The residue from these dark fermentation vessels will be used as a feedstock in a 40,000 litre anaerobic digestion system, and the methane from that will be put into a CHP system, and the liquid portion, along with captured carbon, will be used to generate algae and cyanobacteria. So these are the various work packages that we have, and I'll talk you through who's doing what within them. So the BDC, University of Leeds and Cyanocapture are all involved in the lab scale R&D work. So really trying to pin down as much as we can at small scale before we get to the demonstration scale building and testing. This will take place at the Maltings Organic Treatment Site, which is located just out towards Leeds, near Leeds. Aardvark are covering the planning and permitting of the demonstration site, as well as the enabling works at site. And then we have Cube Renewables, who are involved with coordinating the design, build and installation of things like the dark fermentation systems, the anaerobic digestion, feedstock supply, all the mixing, and material transfer and so on. And then we have University of Leeds and Cyanide Capture looking at the design and build of their various carbon capture systems. We'll be undertaking a 1,000-hour continuous function performance testing phase. And during that time, we'll also be looking at hydrogen cleanup and CO2 separation. Running alongside all of this practical work, physical work, we've got NNFCC and CM90 looking at things like supply chain, market engagement, and then a more developed LCA and TEA, and also looking at any regulatory and policy considerations we need to make for taking this forward as a commercial venture. We have AB Agri, who are and have been and will continue to be supporting the analysis. As you might appreciate, there's a fair bit we need to do to understand this system and to ensure we're getting the most out of it. And then finally, we have Ramboll, who are taking care of the engineering, project management, and the site integration, including various components like the HAZOP report and so on. As Penny said, BDC is providing the project management for the whole project. And with that, I will close and hand over to my colleague, Alex Dukes, who is also at the BDC, who will tell us more about the lab scale activities.
Thanks, Debs. Over to you, Alex.
Okay, hi everyone. Just wait for this to catch up. Okay, there we go. Okay, so hi, I'm Alex Dukes. I'm a senior technologist at the Biorenewables Development Centre, and I've got 10 minutes to have a quick run through of a small part of our project activities. The BDC is responsible for developing and optimising the dark fermentation and anaerobic digestion to fit this biorefinery concept. So as the project's been about generating hydrogen, we focus more on the dark fermentation. Also, the dark fermentation has a lower TRL, so we've had a bit more work to do there than with the anaerobic digestion. So to develop our dark fermentation, we focused on 4 areas. So hydrogen producing inoculum. We needed a microbial culture that would generate hydrogen, not methane, and break down waste materials. Material for anaerobic digestion. So we also needed a residue left after the dark fermentation that would go into AD and produce methane. Optimized process conditions, so giving suitable residency time and enough hydrogen to be commercially viable. And we needed feedstocks. So this included finding feedstocks, volumes, treating them with either enzymes or other means. But as we're limited on time today, I'm going to focus more on the hydrogen producing inoculum. So this is our basic method for developing the dark fermentation. We started with the inoculum, testing different treatment conditions, and then running batch fermentation experiments using the hydrogen, total hydrogen involved, and the hydrogen percentage of gas as success measures. We can vary process conditions such as temperature, starting pH, additives, loading rate. And once the inoculum was finalized, we then started looking at different feedstocks and enzyme treatments, which then again went into the batch system. Once we sort of optimized the batch system fermentation, we then moved on to the 7-litre continuous systems, which are more representative of what we'll have at the demo plant and real world situations. So for the inoculum, so our aims, increasing hydrogen content, so higher percentage of hydrogen the biogas produced makes better profitability and easier downstream processing. We needed a mixed culture that was capable of breaking down materials and then driving towards hydrogen production. Reducing methanogens, so the methanogens will just use up the hydrogen once it's produced, so we wanted to reduce the level of them in the culture as much as possible. Ideally, it would be nice to have a culture that could be fed a wide range of feedstocks, just to increase commercial potential and uses. So rate of conversion, that's kind of been a little bit of a sticking point. So if our conversion is too fast, then we risk flushing away our microbial culture. But if it takes too long, then obviously it reduces the profitability. We needed a culture that was stable. So when I say stable, I mean that once we've treated it, being able to either store it for a short period, how fast do we have to use it after we've treated it, and also repeatability. So being able to do this again and again and gain similar results. So the first thing we looked at was sources of our culture. So we looked at farm digestate. So we initially prioritized food waste actually, because we assumed that the feedstock for food waste would be a closer match. And we also have a food waste plant pretty much next door to where we'll be building the demo site, so it logistically made sense. We also looked at farm digestate. It's easy to get hold of, and we had quite a few contacts we'd worked with before. Plant-based digestate is still one that we are waiting to try later in the project, but the intention there is that if we get something that digests more of the lignocellulosic materials, we'll have better chances of producing hydrogen from these materials. And sewage sludge is something that we're now working with, and I'll talk to you a bit more. So the methods that we looked at, so chemical addition. So basically we did the literature review, came up with a few different options, and some of them we were able to rule out quite fast. So chemical addition, once we looked at the actual cost of adding in these chemicals continuously, it ruled out that. pH control, we tested in the lab, but we had a lot of foaming issues once we looked at this, and we didn't get great control of methanogens. Cavitation and ozone, so we had a trial with this, and we did get reasonable results, but again, cost was prohibitive. Heat and microwave were the two that we knew would be better. So heat we've worked with quite a bit, and microwave we'll look at later in the project. So in phase 1, we started with autoclave treatments. So the University of Leeds had already done a little bit of work here. So we were able to narrow down our heat and time quite quickly. And we got an inoculum that worked really well. But the difference was in phase 1, we were working on much smaller volumes. So when we came to phase 2, we first tested this with food waste digestate, but we had very, very little hydrogen produced. And when we looked into this a bit further, there actually wasn't a great deal in there to produce hydrogen to start with. So we were working with an inoculum that just wasn't ideal for this process. We then moved on to farm digestate, which gave promising results, but we had a massive amount of variability. So this little graph here, so batch 1 and batch 2, these were done sequentially. So one went into the autoclave, came out, second batch went into the autoclave, and then we had completely different results. So one produced hydrogen, no methane, one produced a lot of methane and barely any hydrogen. So the only difference between these two batches was that they'd gone in just slightly, one had gone in slightly later than the other. But obviously that showed that this just wasn't going to be a method that would work. One thing as well with the autoclave that we used is it wasn't stirred, so we had no real control over temperature in the middle of the material compared to on the outside. So we then started looking at different heat treatments. So we moved on to an AFEX treatment. So this is just a 100-litre high temperature vessel. It's stirred and it has a temperature probe sat in the material, so we could see that everything had got to temperature. And we started in basically the same range of temperatures that we'd use with the autoclave, so 90 to 120 degrees. And we started getting some really good results. Also, best thing about the results was that they were really repeatable. Once we did this again and again, we got the same results. So that was a big improvement on the autoclave treatments. We then looked at dark fermentation with these inoculums, and we used basically glucose or food waste soup, and we were producing quite good hydrogen yields. We also found that the material was really stable, so we literally could sit this at room temperature, and our increase in methane was very, very little. The microbial analysis that we carried out as well showed that the level of methanogens was at 3%, and that didn't increase over time, so it did stay stable. So once we had our inoculum in place, we could start looking at different feedstocks. So this is just a little snapshot of some of the materials that we've looked at. So straight away, the paper manufacturing waste jumps out. So that waste was treated with enzymes. but we were getting really, really high yields. So this is based on milliliters of hydrogen per gram of volatile solids. We've also been looking at food waste soup, so typically soup that goes to anaerobic digesters, alternative protein production waste, and we've also been working with this blend of materials from Maltings Organics. So Maltings Organics is where the demo site will be, so they'll be supplying the feedstock for the demo trial. And with the blend of materials, so it's more waste that don't go into AD, so they are genuine waste materials. But we found that actually, if we just change the inoculum to sewage sludge, which is sort of a recent thing, we were able to increase the hydrogen to 184 milliliters of hydrogen per gram of volatile solids. So just that small change with the sewage sludge treated in the same way as we were treating the farm digestate has allowed us to boost the hydrogen a bit further. So next steps for us, so move from batch experiments to continuous for the demo plant feedstocks, continue testing other feedstocks at batch scale. And also we'll be looking at microwave treatments instead of the current heat treatment. So the idea behind this is that it will be cheaper to run. And also if we ever need to produce further inoculum, so if say the demo plant suddenly drops and doesn't produce hydrogen, it'll be a lot easier for us to work up further inoculum. And these pictures are just a little snapshot of everybody at the team here who's been working really hard on this. So thank you.
Thanks, Alex. Okay, now going to hand over to James, who's going to talk a bit more about the equipment and the kit.
Hi, everybody. Good morning. Penny, I haven't quite got control of the screen yet.
It took about 30 seconds for mine as well. It did just eventually come.
Okay.
And you've not got your camera on, James? I don't know.
Hang on. No, not working.
Yep, you've got control.
Okay, thank you. Okay, so quick introduction into Cube Renewables. We are, hang on, we're going backwards a bit there. We're a specialist in small-scale biogas production. So we look for specific organic waste streams and look at fugitive methane gas capture from a various range of feedstocks. We've been going since around about 2012. We came out of a contract with the MOD to look at remote sources of foodstuffs, so organic waste streams in forward operating bases for the UK military. We partnered up with a specialist engineering company, so all of the kit for the H2Boost project is being designed manufactured in our production facility down in Plumpton in the West Country. So we're a West Country based business. In 2021, we also took on an investment from a large waste management platform in Singapore, which allowed us to scale up. So we currently run projects across the world, anywhere from sort of Canada, North America. We do a huge amount of work in India and Southeast Asia. A very quick introduction. So these are some of our some of the products that there's a range of technologies that we roll out. So from very basic containerized AD systems to more deployable units, we have a whole range of CHP generators that we manufacture and modify in-house. We have landfill gas capture projects running in India at the moment with very large landfill remediation partners that we have there. We have a dry digesting technology that's running on rice straw and other agricultural residues. And we do a lot with farming in terms of slurry, gas capture from slurry and air quality pollution. So just moving on, in terms of our sort of background in specialist trial works, we do a huge amount of design and manufacture bespoke systems. So we have several very large clients around the world who are looking at, I mean, we work with quite a lot of oil companies who are trying to diversify out of fossil fuels and look at renewable energy sources. So we're very used to designing and manufacturing and installing really specialist kit that can look across a whole range of different parameters. And that's why we were really lucky to be involved with H2Boost in phase 1. And obviously then into phase 2 for the actual the application, the trial system that we're putting in to the Maltings in Yorkshire. So a little bit around what we're trying to do. So we're trying to follow our modular approach to rolling out technologies and kit. So we're looking at a container-based biohydrogen generation system. You've got the whole background of the sort of the where we're trying to go with this with the project. So we're following the modular approach that we've always had. So it's all complete factory built and commissioned. The design has had to be very agile. We're working with some really specialist partners who are bringing in very specialist technologies. And it's also, it's a trial, so we're all learning as we go along. So the design has had to be incredibly agile. We are looking to put into the plug and play controls. We are looking for it to be as remote as possible, with sort of minimum disruption and on-site installation. Cube has always taken a huge amount of data off all of our units. So we process data from various data sets around the systems. And that's a key part of being able to report back to the team. So again, it's around about that sort of integration with some of the specialist technologies that we're looking at. So in terms of upgrading, carbon sequestration, etc. So we've got a really good track record at that. And as you can see here, these are some of the photographs of the the units in production. To show you where we are now, so obviously we're talking about agile design. So that's the P&ID at the top here. So I mean, we can take you through that in a lot more detail if required. We certainly don't have time for it on this call. So really, so moving from the P&ID to the rendered design here to the build down in Clumpton in the West Country as we are now. So to take you through this, it's probably best just to go in stages really. So if you look here, and it reflects, the rendered model reflects the P&ID above. So you've got on the left here, you've got these two containers here, which is all around hydrogen upgrading systems. So within that, we've got two streams, but we're upgrading hydrogen up to 99.9% purity. The small unit on the left here is around the low pressure H2 storage system. So we're not converting to anything higher on this project at this time. So there's that's your storage systems here. These units here, so within this you've got the CHP generator, which is provided by us. You've got a biogas boiler in there, and you've also got two gas control pods. So cleaning out so gas treatment, cleaning out the hydrogen sulfide. One of those is obviously for biogas, and one of those is for hydrogen. This bit here is where it starts to get really interesting. So you've got within this, so the first container here contains our control system. So that's full SCADA system. It also contains a hot water and air control system as well. The middle unit here is the dark fermenter. So you can see pressure relief valves here at the top. So again, the two tanks that Alex was talking about recently are integrated into that container in a sort of safe and easy to manage system. And then on the right here, so in that container there, so the third in that row, you've got the full pasteurization system. So obviously that's important when we're dealing with things like food waste. At the back here, so you've got a storage tank, so that's effectively incoming digestate, incoming feedstock storage. And at the front of that here, you've got the steam boiler and the chillers for the pasteurizer. So these are all linked back into the pasteurization container. And then moving on to the container on the right, you've then got our standard biogas system. So as Alex said, that's it's actually got a storage facility of around 43 cubic meters. And then at the front there, you've got your hydrogen flare. So that all reflects and it's all built out. And I'll just run through some photographs in a minute. Just to flag up the only items that aren't shown on there. So we put a blend tank on the front of it to be able to introduce feedstock according to our software systems. There's a couple of water tanks and flush tanks that aren't on here, but otherwise everything that's going up to the maltings is all listed out here and identified. So just moving on to the next slide. I think the host has just been changed.
No, you're still controlling, James. There you go.
Okay, so there's just that's the actual kit in build as it is now. It's nice to have a finally a bit of blue sky above Devon. So that was taken on the sort of one day of the year that it wasn't absolutely throwing it down with rain. So on the left there, you've got your anaerobic digester unit. So that's obviously biogas production. You can see all the scaffolding up here that houses all the air tank or air pipes and all the electrics for the build. In the middle here, you've got, so this is obviously your dark fermenter, PRVs at the top here. So the tank's integrated into the container systems, and you can see that here. So we've got room down the side to be able to access and maintain tanks there. And then we mentioned the control room here. That was sort of halfway through the build. So all the electrics, all the control panels, everything's actually linked up and ready to go. There's a little bit more in terms of the wiring, the last bits of wiring to do. In the middle here, you've got the biogas treatment pods. So as we explained, for both, so this is one for the biogas to take out the hydrogen sulfide. And on the right there, you've just got some piping and some of the engineering work.
Great. Thanks, James.
Yeah. Okay.
Brilliant. Thank you. I'm now going to hand over to Miller at the University of Leeds, who is going to talk through the carbon capture.
Yeah, thank you very much. Apologies, I don't know why the camera has been switched off. So I'm going to go through the slide. So good morning, everybody. My name is Miller Alonso Camargo Valero. I am Associate Professor at the University of Leeds. I am leading the Bioresource Systems Research Group in the School of Civil Engineering. In the past 15 years, we've been focusing on the optimization of different cycles. When I'm talking about cycles, key elements like carbon, nitrogen, phosphorus, and obviously water in waste management systems. We try to bring more sustainable approaches and contribute to net zero targets. So what we are doing, or we are based in engineering, in an engineering department, we took a step back to see what is happening within the different processes that they are linked to waste management. And we try to identify the basic principles that can help us to modify or to change current processes to achieve bigger goals like net zero. So that means that we go from fundamental research all the way through to process development. And in this particular project, we have the great opportunity to translate that into a pilot system that we will test the feasibility of implementing carbon capture processes linked to the use of microalgae or photosynthetic organisms in general. As I mentioned before, we started some years ago, and the main challenge that we took was the recovery of nutrients from wastewater. And we identified that the current approaches, they didn't promote the recovery of nitrogen and phosphorus in particular. They were very good at doing control of those nutrients. And as a consequence of that, the amount of energy that is required is very high, and still is very high for many systems in the UK for wastewater treatment. So we start to look at different options, and we identify microalgae as a suitable organism to test and to identify how we can increase the capacity of recovering nitrogen and phosphorus. So we, as I said, we took a step back and we tried to identify exactly what is happening with the processes, the mechanisms controlling the sensing of phosphorus outside the cell, how that phosphorus is transported across the membrane, and how that phosphorus is accumulated inside the cells through the formation of polyphosphate crystals. So this work is based on research funded by the BBSRC and in collaboration with Professor Alison Baker, also from the University of Leeds. So at that point, what we saw was the great opportunity to contribute to net zero to agriculture and to waste, particularly wastewater sectors. At the moment, these two sectors, when we combine these two, they contribute to about 12% of the greenhouse gas emissions in the UK. When we try to look at that figure in more detail, we found that about a third of that is linked to CO2, and is linked in agriculture, for instance, probably a half of that is linked to the use of fossil fuels, and the other half is embedded in the supply of feedstock and fertilizers, which means that activities like heating, cooling, power supply, and transport, which is common to all different sectors, by moving from fossil fuels into our renewable sources, is going to make only a small contribution towards net zero targets for that industry. So when we look at waste management in general, what we have is that current practices, they promote the release of greenhouse gases like methane and nitrous oxide. Just to give an example, the agriculture sector on its own contributes to 48% of methane emissions and 64% of nitrous oxide emissions in the UK. So the current practices, they are not the most sustainable. And for that reason, by providing the option to capture those nutrients, we can reduce those emissions in the sector. Obviously, here we have another component that is very crucial, microalgae or cyanobacteria as well. They do photosynthesis, which means that they can use inorganic carbon dioxide, for growth. And in this case, we can bring these two together by recovering nutrients from nutrient-rich waste, like digestate or wastewaters. But at the same time, we can capture CO2 that will be incorporated into the biomass. So that's the concept, and the development of that understanding of improving our understanding of how we can capture and uptake nutrients using microalgae. That was the driver that bring key developments or scientific knowledge that we are applying to this project. So if we look at the composition of the microalgae, typical composition, 50% that is carbon. As I said, they do photosynthesis. That means that they use inorganic carbon, CO2, as a carbon source, but also they can incorporate nitrogen and phosphorus. And that was the key element for our development, focusing on the limiting nutrient that is phosphorus. And we can see that on the normal culture conditions, microalgae is able to accumulate something between 1 and 2% of phosphorus, which is used for DNA production and energy storage as well. That's what we have found. So in nature, when we have low concentration of phosphorus in a river or in a lake, microalgae start, let's say, to panic because there is no enough nutrients, and start to bring as much phosphorus within the cell and store it, and store that for cell maintenance and reproduction and cell division. So store that just simply because has been into a stress environment. And for that reason, it was very difficult to combine two key elements, which is biomass growth that if we grow biomass at large, very fast, we can capture more CO2, but we cannot capture nutrients at the same pace, because if you want to grow that very fast, you also need nutrients. So you need a nutrient-rich environment. So that was the 2 points, that if you want to have high biomass, you need nutrient-rich environment, but we are not going to be very efficient in capturing the nutrients. So we were looking at that in more detail. As I said, from the engineering point of view, we were modifying different aspects in terms of the operation of the reactors, modifying environmental conditions, and we were able to increase the concentration of phosphorus from 1%. So here, I don't know if you can see my cursor, if that is moving or not, but let me see if we can do, probably not. If we can see here to the right side of the top right side, we can see a standard cell of microalgae that has this black dot. That is how we stain the polyphosphate, and that is about 1%. And we modify environmental conditions, we can have up to 4% with a larger accumulation of polyphosphate. But we really didn't understand exactly what was happening. So that's why we partnered with colleagues in biology to try to understand this much better. And we were able to enhance the content of phosphorus and the growth at the same time, up to 7% of phosphorus inside the cell, by doing some modifications that give us also clues about how we can translate this into a full scale process. I'm trying to change the slide, but it's not working. Yeah, have it here. So we have that option to scale things up, but we need to understand much better in terms of how we can translate lab data into pilot scale systems. And I know that probably you have heard about microalgae and the limitations of using microalgae. Using sunlight is probably the cheapest option, and we are limited by weather conditions here in the UK. But also the other limiting factor is how fast they grow, and that in many cases requires that the residence times, the order of days for doing that. So here to the left, what we have is some results that they are showing the response of under specific conditions for the growth that we conducted in our lab. So we have here this black line at the top that is giving us our control. So when we give about a week, we were not able to remove the nutrients, in particular the nitrogen. But when we modify the microorganisms, where we're using genetically modified Chlamydomonas, the microalgae model organism that we were using, they were able to remove phosphorus to below detection limits in 3.5 days, which is for us was very important, because by using this organism that was modified to maximize the transport of the phosphorus from outside the cell to inside, we identify what was the mechanism that was controlling that element. Obviously, the release of genetically modified organisms in the environment, we want to avoid that. But now that we know the mechanism, we were able to translate operational conditions to replicate what was happening there, and have environmental conditions in the culture that trigger the specific genes that accelerate the transport of phosphorus from outside the media to inside the cell. So to the right here in this graph, we have a cluster of results. So the ones that we have, that they are a long flat line at the bottom, that is the combination of different strains that we tested, that they are not available in native species, that they are available, that they haven't been modified at all, and they were able to reduce the concentration of phosphorus below detection limit in 12 hours, which is something that for us is a game changer in terms of how we can translate this into real life conditions. So based on that, what we're trying to do now is to replicate this using the different nutrient sources that we have in our project. And for the cultivation of microalgae, yes, we need nitrogen, we need phosphorus, but also we need CO2. So within our project, we, as Alex mentioned before, we have the dark fermentation unit, and that will produce hydrogen, and it's a mix between hydrogen and CO2. And we are going to have a system that will upgrade that CO2 to the quality that is required for transport. So in that process, we are going to have some CO2 that, or probably a mix that is going to come up with the cleaning unit. And this is one of the sources of CO2 that we can have. Remember, if we think about how our country needs to report greenhouse gas emissions using the IPCC methodology, which is set by United Nations, CO2 that comes from any biological process is labeled as biogenic CO2, and that doesn't count into the emissions. But if you have a biological process that releases methane or nitrous oxide, they do count within the greenhouse gas emissions. And that, to me, makes a huge difference between the use of carbon capture and sequestration processes and what we are proposing, which is carbon capture and the utilization of that carbon in other sectors that ultimately will reduce the use of fossil fuels. And I'm going to expand this in a little while. But just to mention where we can get the CO2 from, and this is one source. The other source that we have is the anaerobic digestion, and the anaerobic digestion is very key for this process. Yes, we can optimize the production of hydrogen, which is the target, but ultimately we are modifying a waste management system. And if we are using waste, we need to comply with legislation in terms of how the waste is processed. And to do that, we need to further treat and stabilize that organic matter, that organic waste. And for that reason, anaerobic digestion is there with the benefit that will produce the additional energy that is required to run the system. And hopefully, if the project goes as expected, we are going to have a system that is not going to need extra energy sources to run. So going back to anaerobic digestion, as you know, that produces biogas, that is methane and CO2, and that is another source of CO2. If we send that to a unit to produce energy, we are going to have that also that source of CO2 for growing microalgae. At some point, we need to bring the microalgae, and that means that we need the CO2 that can come from burning the biogas, or can come from the cleanup unit for the hydrogen. But also we need the nitrogen and phosphorus source, and that comes from the digestate. So the digestate liquor, the liquid that can be separated from the digestate coming from the anaerobic digestion, is rich in nutrients. And that's the source of nutrients that we are going to have for the growth of the microalgae. So going back to the composition of microalgae, nitrogen is about 10%. Phosphorus hopefully will be higher than the standard, which is 1 to 2. And that will produce a product, which is the biomass, rich obviously in carbon, and is going to have the nitrogen and phosphorus. If we follow the carbon capture and storage, that means that we need to be sure that that carbon stays in that form, in a solid form, for over 100 years. So that is the sequestration part of the carbon that is within the guidelines from the IPCC. If we do that, we can comply with those requirements to say that is the carbon that has been released in the system is stored, and for that reason, we don't count those emissions. But as I said before, this works very well when the original CO2 comes from fossil fuels. So that means that needs to be stored or used in a different way. In this case, the CO2 that we're using comes from a biological process. And for that reason, we can have other opportunities. If we're going to store the biomass, the option that we are proposing is taking that biomass, produce biochar, and then store that in a safe way so that carbon stays in the soil or stay in the location where that is going to be stored. But the alternative that we are proposing is that once we have captured that carbon, along with that, we are going to have nitrogen and phosphorus, which is one of the, it is very vital for agriculture. Nitrogen and phosphorus is part of the fertilizers that we need for the production of food. And for that reason, if we replace the use of fossil fuels in the production of industrial fertilizers, this project will give the opportunity to recycle those nutrients without affecting the commitments in terms of carbon emissions. And that ultimately will help to reduce those nitrous oxide emissions or additional emissions, greenhouse gas emissions coming from the production of fertilizers. And that is probably the exciting part that we want to bring here to the project. Okay, we have our expectation in how we are going to run our pilot system. This is in a shipping container similar to the ones that was presented by James from Cube. And that's where we're going to have our system is going to be running with the CO2 that is produced on site, and the nutrients will come from the digestate liquor that is also produced on site. So we are self-sufficient in terms of the nutrients that is required, and obviously the water is already there because the digestate liquor is diluted. Yeah, we are very excited that we are at this point in the project, and just the last part is to this work is a result of a combined effort with everybody in our team. Not everybody that is in the project is in this picture. Hopefully for the next workshop, we will update that picture. And that's it from the University of Leeds. Thanks very much.
Thanks, Miller. I'm now going to hand over to David at Sinocapture. Oops. Miller, I think you're still controlling. Just a minute.
I don't know what to do. I think the
That's
change
right. Do
was
nothing.
done by someone else.
Yep. David, you should be able to get control now.
Thank you, Penny. Thanks, Miller. Good morning, everyone. I'm David Evans from Sinocapture, and we're a startup based in Oxford. And Sinocapture was founded in 2021, and since then we've won an X Prize and in 21, and we won the Shell New Energy Challenge in 2022. And we're really excited to be part of H2Boost and to work especially with BDC and University of Leeds. I'm going to kind of talk through our approach as a company to how we see sort of carbon capture. You'll hear me talk a lot about energy and cost, and then kind of moving forward then into sort of what we're actually doing in H2Boost. So first of all, we're using cyanobacteria. So you've heard from Miller, they're focusing more on microalgae, and cyanobacteria were the world's first photosynthetic organism, and actually about half the world's oxygen today is still produced by them. So they've become incredibly efficient over many hundreds of millions of years at fixing CO2, and they produce lots of interesting products as well. So we've been on a journey over the last sort of couple of years in terms of how we actually get hopefully the economics of our carbon capture system to actually work. Just taking a step back from that, I really like this chart from The Economist, which shows as much as we need a lot of renewables and to reduce our energy demands, we need carbon removal technologies. And as Miller said there, biogenic CO2 sources are a very high quality form of carbon removal when we can capture that CO2. So that's kind of what we strive to focus on, and that's sort of why we really like this being part of this biohydrogen pilot. So just to touch on the carbon capture space right now and the problems with it, is that frankly, it's just too energy intensive and too costly. So we're looking at kind of carbon prices of over $400 a ton for captured CO2 currently. And just in terms of energy use, you're looking at sort of, significant portions of global energy supplies to capture the CO2 we need to hit our IPCC targets. And I guess our view is that the renewables and electricity should be going as much as possible into decarbonising current electricity demands and supplies, rather than for kind of CO2 capture. And then just to touch on kind of BECCS and biochar is that again, everyone's fighting or looking to fight over utilising biomass through growing feedstocks to then be used for either energy production or CO2 capture. So that's why using waste streams, hence we're doing in this project, is such a valuable way to not have any opportunity cost associated with land use. So this chart here, which has a lot of big circles on it, just kind of shows you the state of the sort of carbon capture market right now. This is the voluntary carbon capture market, and it's a very nascent market, but we can kind of see on the charts that, broadly speaking, is that most of these carbon capture purchases, especially the large ones, have been in the several hundreds of dollars a ton, if not more. And we just think this isn't sustainable, as we're starting to see as an approach to kind of hitting our net zero goals. So what are we doing about that? So as I said, we're using cyanobacteria, and for H2Boost, we're just doing, as I'll get onto later, just some basic screening selections for strains. But at Cyanocapture, we also focus on genetic engineering, and that's both in terms of a full CRISPR program and also in terms of directed evolution as well. But we're using these highly efficient natural systems to try and fix CO2. So what does our process mean? I mean, Miller kind of touched on this as well, but broadly speaking, is we're taking flue gas. So for H2Boost, this is from the CHP, and the digestate from the AD, putting it into photobioreactors, which contain our cyanobacteria, and then they rapidly fix CO2, producing more of themselves. And then we're looking for this project into pyrolysis to lock away that CO2. But then, as I explained, in terms of our economics, we have a range of different options as well in terms of looking at biocrude, and then something we're exploring in the future is also looking at graphite, because the properties of cyanobacterial biomass are very interesting in terms of graphite for batteries and EVs. So what does our modelling kind of tell us? So for a 10,000 tonne site, we're looking at about just under 11 million pounds worth in terms of capex, and then our LCOC. So that's basically taking all our costs and then minusing those costs off our revenues. Means that we actually end up being profitable even without carbon credits, which we think is a hugely important aspect to do, as the carbon capture market, especially in the voluntary sector, is extremely turbulent and nascent. And while doing this in an outdoor setting, we think we have really, really low energy and electricity requirements as well, with super high quality measuring of the carbon that we're fixing too. So how are we doing that in terms of the cost? So we've been on a real journey certainly over the last year, 18 months. We came into this thinking that we'd sell carbon credits, or be as a kind of a contractor as part of this part of H2Boost, and we realized actually the numbers just didn't really add up on the carbon capture side. So as kind of, again, Miller said, and I'm sure Chris will touch on as well, we're looking at to make the economics of this actually work, which is so key in terms of carbon capture, is that we're looking at firstly algal sales. So as Miller said, whether that be for fertilizers and feeds, but then also torrefaction, which is another form of pyrolysis. I won't go into the reasons exactly we're looking at that, to refine the rest of the algae, so that then that carbon is locked away for a period. And this means that we hope that our model actually makes it into the black. Okay, and then so what are we doing for H2Boost? So again, as kind of Miller said, we've been working very closely with the University of Leeds in terms of looking at a wide variety of strains. and then sort of filtering those down to the ones that are most suitable for this project in terms of looking at the ones that are growing best within the AD digestate. That means basically growing up the sort of the scale of the strains that we're testing. And then all the way to now, we're doing our inoculum grow up in 15-litre bags within our lab, and then moving into inoculating the system up at the malting site very, very soon. Just on the biology, I mean, it's not been all clear sailing. The literature says that AD has been used for growth for cyanobacteria and microalgae. We've had to adjust to some new systems. So we're working with the BDC to reduce the turbidity of the digestate samples. So we've been flocculating that, and that's had a bit of an impact on growth. But then we've sort of seen that actually after a few days that subsides. And then we've also seen a degree of sort of toxic compounds for the cyanobacteria, and we've sort of narrowed that down to formic and acetic acid. And so we've come up with this sort of middle ground of diluting down the digestate and supplementing it with additional nutrients. And you can see some nice green-looking flasks there in the box in the bottom of the screen, and that's what we're looking forward to hopefully having success with up at the maltings. And then in terms of our system, so we're kind of, I suppose, the most experimental, cutting edge sort of experimental and novel, because we are new part of this system. So we have an indoor system to kind of help control a certain number of factors. So you can see in figure 1, that's our photobioreactor, all assembled, ready to be installed in a container. So again, we're all in containers for this project. And then we have a designated light plan, which you can see in the bottom left, to try and help us optimize our growth in there. And then again, similar to kind of Miller, we have a variety of sort of tanks in terms of mixing, flocculation, etc., which the inoculum is pumped through, so we can optimize the media mix, and then it's pumped through all the tubes, and hopefully we get some really nice growth and we can capture lots of CO2 as a result. And that's it from me. Thank you very much.
Great. Thank you very much, David. We're now down to the last 2 presentations, for those of you still with us. So we're going to have Chris Corner next, who is going to talk more on the policy and the techno assessment. Chris, you should have control.
Yet. Just wait for it to come through.
Yep.
While we wait for that, so I'll just say hi, morning, I'm Chris Corner. As Penny says, I'm from CM90. I've got about 25 years of renewable development experience, and including biodiesel, bioethanol, plastic recycling, biomass, recently large-scale batteries. Helped BDC a few times in the past, but at this point we're looking at the techno-economic assessment, and we're going to talk a little bit about supply chain and policy today. I still don't have control, Penny.
You do now.
I do now. Yeah, it's just come up. Very good. So just to sort of outline techno-economic assessment and how this relates to this project, and typically this comes together in the second half of any project, but is entirely data driven. So we need all the information from the project, and to have a structure at the beginning makes sense, so people know what we're looking for to bring this together at the end. The objective really, particularly in this case, is about determining the deployment potential for H2Boost, and so that's a technical question, but the commercial question is, can you do it in a sort of investable fashion? So albeit it's data driven, the next step is to get money from venture capitalists for scale up, for example, but in an operational plant, or to get to an operational plant, you need development funding, and you need money from the likes of a private equity or pension fund. And all of those money parties are looking for as much certainty as they can get, or a large opportunity with risk. but everybody wants to get rid of risk. So that's the job of the project. So in this case, we're looking for data right the way through, and not only what performance can be achieved, but it's what the future potential performance might be. And as James mentioned earlier, the plant configuration is what it is today, but there may be a different configuration in the future, which is more compelling. But we won't know how to assess that until we've got more data from the various elements of the project. So at this stage, the TEA can look at building a picture about feedstocks and products and whether there's appetite for contractability. We can start to look at where that fits into the hydrogen supply chain, which has its own sort of technical issues. But ultimately, all of that will drive consideration of what the actual commercial scale might be. So, for example, that we just heard from Miller and David both mentioning biochar, you can ask yourself, what's the smallest commercial scale of biochar conversion unit, which then says, well, how much algae or cyanobacteria do we have to produce by this process? And then you take that forward right to the feedstock and say, how much feedstock do we need to do that? And then the bit in the middle, which is the plant, can that be done as well? So all of those elements are what we'll be considering in the second half of this project. The extra factor that's sitting on top is policy. That drives not only investment direction but compliance requirements. In this case, the compliance requirement is it has to be a low emission project, and so this element, the TEA, interfaces with all elements of the whole project, including a very close link with the life cycle analysis, which is mass and energy balance focused. So in the round, that's a lot of elements to pull together, but it will come together in the second half, I'm sure. So just looking at the supply chain, we've got feedstock and products, and I'll just say that this element's done with support from BDC as well. So we're working together to do elements of the supply chain work. We've considered the scale of the plant is really, for now, what was the phase 1 scale. So we had a 47,000 tonne a year feedstock in-feed plant. The main focus of attention is on the remaining 2 promising feedstocks, which is food waste and then paper and card waste. The findings for this are done in 2 ways. One is a more granular view that was done from what was done in phase 1. So trying to get down to exactly what we could contract. And then secondly, actually engaging with the potential suppliers, which is what's happened over the last few months. So as you can see here, we've got a thick end of 9 million tonnes of material available in theory. In practice, that's all going somewhere today. The 4.8 million tonnes into AD plants, which is the focus of the project, to back up to an AD plant, is already contracted. So the question of can you contract that is yes. Can we contract that? Unclear at the moment, but we'll come to that. But we do expect additional volumes to come out because there's change in the legislation for segregated waste. In other words, we're all going to have another dustbin at home and put our food waste. If you haven't got one already, we'll all have one by 2026. And that will wash out more material that can be processed. And currently that's being targeted at AD, but arguably dark fermentation is a form of AD. So we may well be able to get the government goodwill behind that towards this project. That is an area to examine. In terms of paper and card waste, that already goes to places for recycling. There's been up to an 80% recycling rate on paper and card. It's dropped off a bit in the last couple of years for various reasons, but that will come back. So this project is looking at the lower grade element of that paper and card waste and what might come out of the back of, for example, a recycling mill or a paper mill. And so those numbers will not be as large in the end, but we don't know yet what sort of scale they will be at. In terms of the economics of that, market prices, as you see there, are sort of anything from 29 to a gate fee of £1 for food waste, and you don't see a gate fee for paper and card. This is the lowest grade at £5 to £50 a tonne. So that all has to wash through to how much will the hydrogen cost ultimately. And the graphs show us sort of some geographic spread, which will map onto the AD plants, for example, that Deb's put up earlier. But you can see it's more about where material isn't rather than where it is. So food waste is everywhere, which strangely enough is not in the West Midlands, which must mean that is dispatched to other regions for these aggregation points. Whereas paper and card is very definitely east of England, northwest and southeast in terms of the aggregation points. So that's very helpful to us because we can address those. In terms of engagement with the market, really, that's early days in terms of what we can try and ask to contract, because we haven't got all the feedback from the project yet, but we started it. And it's unlikely we'll have a single contract. And if we do have a single contract, we will have to manage the material quite closely on plant, so that'll affect the plant design to end up with a mix that's suitable for the process. Now that's very similar to what we see at the Maltings and AB Agri and other sites, but not. It really will dictate how we contract the feedstock. So let's now look at the other end of the process, which is products. Again, assumed scale, as I said, 70 tons a year of hydrogen there. I've put 140 as the upper end, which is kind of just a bit north of what's being achieved if I extrapolate the laboratory results that have occurred in the last few months. So that's really promising. Focusing on hydrogen particularly first, there's a government target of 10 gigawatts. Hard to see what that actually means. What it means in my assessment is in terawatt hours. I'm trying to find my figures here. It's 460 terawatt hours of hydrogen by 2050. So the plant at 70 tons is about 3 gigawatt hours. So that puts it into context. So where's the hydrogen going to go? We're focused on transport. We have to look at other alternatives if that doesn't come to pass. But gas grid injection is not approved yet in the UK. It's on the cards, even though we hear lots about HyNet in the northwest and so on. It's not approved yet. And the transport use is very nascent. If you want to find an offtake contract for hydrogen into vehicles, that's going to be quite tricky. But the quality, we hear this 5 nines, it's called, the 99.999% pure is what they want. In reality, it's driven by sub-factors like how much sulfur's in there and a few other elements. But these are things that the project will have to consider in detail in terms of its deployment. And there is, in sticking with mobility rather than vehicles and fuel cells, there was a suggestion from a number of industry players that we would be good to supply the hydrogen into sustainable aviation fuel, which is now, I don't think it's in force yet. There's a mandate coming forward for inclusion of that. So that is a real market. Then the last bit of the problem is the big one is how do you get it from where you are to where it's going to be consumed? The logistics and storage part of the UK infrastructure is behind the production of hydrogen at the moment, but it is coming along and it's being supported by government, but we have to take a guess at where we might land. At the other end, which is the whole point of the project, which is carbon capture and carbon reduction, I've focused on biochar here in this slide, recognizing there are other applications for algae and cyanobacteria, but we need to have some figures for the life cycle analysis, and the largest application area where we can get long-term storage is to actually apply it to land. But the UK is behind many other nations in terms of accepting that. So, but it is getting there. It's just when is the question. So in terms of the specifics, we need to see what David and Miller's output will be as to how good that biochar will be. We have some projects out there today doing it, but they are supported by the Net Zero programme, and there's field trials supported by the government, but the Environment Agency will be the determining factor here, and we just keep an eye on that for now. I haven't talked about biomethane other than to say that it has a market, and it's for gas grid injection or for transport fuels. They will remain, but the policy support for AD for new entrants ends in 2028 currently, but there's a consultation out, so it's got a question mark over it as to how we'd engage with the biomethane side commercially. Last slide. I'm not going to go through this in massive detail, but just want to point out that the policy and regulatory influences here are broad, starting with net zero. We've got a strategy, we've got carbon budgets, we've got carbon markets. David talked about the informal markets, because there is only an informal market for the biochar, but the formal markets are for those who are trying to reduce their emissions. Notwithstanding all of that, hydrogen and biohydrogen is very much supported, so we're in the right place. Hydrogen has more evolving policies than you can imagine, but the key one for this project is the hydrogen production business model, which is now the hydrogen allocation round 2 is concluding, and that basically gives projects a low carbon agreement, which gives us money for what we produce. So we will keep an eye on that and bring that in financially in the second half. We've already talked about biomethane and biochar. In terms of regulatory requirements, end of waste presents a potential risk to the project. End of waste is a piece of legislation that makes a waste material stay a waste material until it's not, so that any product that's produced from a waste has to be approved as appropriately not a waste, other than because it just prevents a huge amount of difficulty for businesses to sell their products as a clean product. However, AD is approved for end of waste through its quality protocols, and we're going to examine during the second half of this project how dark fermentation may well be classified as AD under those quality protocols and what the pathway would be for deployment, which would be a big win if we can sort of find out the pathway there. And then the last point, which is with new projects, getting a planning permission can be very difficult, particularly with new technologies, and mainly because there is no regulatory framework for hydrogen currently, other than the fossil type, and planners don't have really a set of rules to work by. So we keep an eye on that. There's some papers out by some lawyers giving advice into the way forward, and we'll engage with them as well. And that's probably enough for now. Thanks, Penny.
Thank you very much, Chris. And then finally, we have David Turley from the NNFCC. I'm going to wrap up the presentations.
Thank you, Penny. Just waiting for it to come through.
Yep.
Hi, yes, Dave Turley here from NNFCC, and just following on from Chris's presentation, as you can imagine, looking at the GHG impacts of hydrogen production, which is a key part of what the sort of whole range of projects about. It's an important factor that we need to look at in terms of looking at how all these sort of integrating pieces that you've been hearing about from the previous discussions come together in terms of how that impacts on or how that might integrate into a wider system impact, as well as impacts on low carbon hydrogen production. We're involved in a number of GHG assessments, both for the anaerobic digestion sector as well as for the The fuels and energy sector do a lot of regulatory GHG sort of carbon counting in that. So it's a useful exercise now to bring this to bear into hydrogen production sections and interests. In phase one, you know, clearly a lot of this, as we say, it's very much dependent on the data we get out of the projects. And the first phase of the study involves in doing quite a bit of sit down with the experts that you've heard from today in terms of getting a better understanding of process transformations, how energy flows through the system, how mass flows through the system, and about hydrogen production that we can get from the various systems. And that was backed up by a bit of work from both Leeds and at the BDC in terms of giving us some limited information in terms of how the process might work at a laboratory scale, and that enabled us to do an initial pass in terms of looking at the greenhouse gas impacts. This second phase, obviously, we're now moving into a much larger scale with sort of demonstration phase work going on the laboratory testing, as Alex was talking about, and obviously into the full scale work that Cube was talking about in terms of really scaling this up to something where we can now start to integrate all those processes together and generate the data that comes out of that to look at this in much more detail. In doing that, there's always a question with greenhouse gas accounting about how we do that and the process we do that with. And clearly we need to make sure we're comparing apples with apples and not apples with bananas, which is a common sort of issue when we're dealing and looking at greenhouse gas accounting. And so we're actually working to a common standard across all the projects that are being funded by DESNZ under this round, and that's the low carbon hydrogen standard. So that really defines the approach and methodology that we have to follow in terms of defining what constitutes low carbon hydrogen, and that's up to a point of production, which we'll come back to. So that means that we can have confidence in the numbers that we get out of this in terms of the greenhouse gas savings that are calculated at the end of this, that in all the projects and certainly any commercial scale hydrogen gets produced in the UK is all being assessed and challenged to the same degree. And that gives us some comparability between all sorts of systems so that we can compare our low carbon hydrogen system with blue hydrogen, with electrolysis, etc., so that we're comparing everything to a common format. One of the things that the low carbon hydrogen standards does require is that we have to, if it's going to be called low carbon hydrogen, then we have to get under that threshold of 20 grams of CO2 per megajoule of hydrogen. And a common point of delivery of that, it must be that it's at least 3 megapascals and 99.9% hydrogen. Again, some of the issues that Chris Corner referred to previously in the techno-economic. This is about, we're getting to a stage where it's ready for deployment and use. So in terms of that assessment, for those of you who are sort of familiar with greenhouse gas accounting, that clearly includes things like scope 1, 2, and 3 inputs. So all energy involved in the system, all electricity purchased and brought into the H2Boost system, as well as all the sort of consumables that are used in that process, wastewater inputs, wastewater outputs, etc., and all nutrients consumed in the fermentation process. So all the emissions associated with that need to be taken into account. The bits that we don't take account of, and that's fairly common in this sort of analysis, is the emissions involved in construction and obviously dismantling at end of life. Those are things that are typically outside scope of what we're looking at. That it excludes the sort of downstream distribution of hydrogen once we've pressurized it. So it really goes up to sort of, if you like, sort of the hydrogen factor gate at the end of the H2 process. And what we also don't account for is emissions in aggregating that waste, and so the household collections and the emissions associated with that are excluded. In common with most sort of processes, the point at which that waste is aggregated is that those wastes are taken as having zero emissions. And of course, we're not using any voluntary carbon offsets within this. This basically means that it's a process that has to stand on its own merit. We're not using any sort of market carbon credit mechanism to offset. So, you know, it's a real measurable impact that we get at the end of the day. Just a sort of schematic of the whole process. So obviously it's starting on the sort of left-hand side in terms of collection and cultivation of processing, going through a range of pre-treatments and pasteurization into the dark fermentation, which sits in the center of this process. And then we have the options in terms of the first phase of that, a very quick and rapid process. We'll go into hydrogen production, and we will be separating the hydrogen from the CO2. The CO2 is going off into an algal system, hopefully to capture most of that CO2, and the biomass can be going to a biochar system. Again, we have the options also of looking at that in a larger scale of taking that into a carbon capture and sequestration process. So again, that's something else we'll look at. What we get in producing that hydrogen through that dark fermentation process is a digestate, which we see coming out in the center here of the process, which itself, okay, we've reduced some of the energy in that material, but it still has the potential to produce biomethane, and certainly the integration of the biomethane production with the hydrogen production is an interesting part of the synergy here in terms of how we're going to look at the process. So the first part of the LCA focuses purely on that the first top blue box there in terms of hydrogen production to that low carbon hydrogen standard. But again, stepping back and looking at it into that whole process we've talked about, and a number of your previous speakers have talked about in terms of how does that integrate with biomethane production. We've got further digestate production from that then goes into land, potentially sort of reducing fertilizer use. And again, similar to the algal sort of byproducts, again, that biochar can go into soil and help back out fossil derived fertilizers. So quite a big range of factors here that can be integrated into that assessment. What do we know from the first phase in terms of our expectations? The first phase, which was, as we say, based on mainly laboratory work, was actually really quite promising. And if we sort of look at that in terms of I've got figures here in kilograms of hydrogen, but if you look at that in terms of kilograms per megajoule, then actually on that left-hand graph, the colored bars above the 0, which is in the middle, shows that actually, based on our best assumptions, we should be under that 20 grams of CO2 per megajoule limit for low carbon hydrogen. So again, it looks promising. And with that algal CO2, if it works to expectations, then again, we should be able to produce hydrogen with a net negative footprint on it. But again, that's what this whole second phase is about, is now looking at verifying those materials and numbers we've used in that first phase process to verify our system. So just rounding off, really, what we're looking at is a cradle to gate assessment of hydrogen production, essentially, of this dark fermentation process to that low carbon hydrogen standard. That'll be embedded in a wider assessment of the impacts of that integration with those other processes that have been talked about. And we'll develop an energy mass balance model that's going to support the work of the techno-economic analysis. And again, thinking about how this technology is going to be deployed at scale. Thanks, Penny.
That's great. Thanks, David. Okay, so I hope that's kind of given everyone a bit of a whistle-stop tour through H2Boost phase 2. And that, you know, that if you have any questions, then please do get in touch. I'm just trying to move the slide up. There you go. We've had some questions come through, which I shall pose to the panel. So, so the first one is probably for you, Alex. Can you see the increase in the efficiency of the biohydrogen production following the inoculum pre-treatment?
Yes, yeah, we can. So we have tested it before being treated, and we don't really get hydrogen yield particularly from it. We see a little bit of trace hydrogen, but it gets used up by methanogens. But depending on the treatment, so sort of above 110 degrees, we were seeing methanogen, methane drop off in our biogas, but then as we sort of increase the temperature and the duration, I think we cause more stress to the hydrogen producers, which a lot of them spore form. So we then actually get a higher bump when we first start, which, yeah, so we do, yes, that's the short answer.
That's fine. That's great. And also, another kind of question on the dark fermentation is, have we tested different conditions for the dark fermentation in terms of HRT, temperature, and how does that affect the biohydrogen yields?
Yeah, so in phase 1 we did a lot of work around starting pH and temperature. So we actually use a slightly higher temperature. So we use 45 degrees at the moment, which we found also helps to inhibit some of the methanogens but increase our hydrogen production. The HRT we'll be looking at a bit more once we're working on our continuous systems and they've stabilized, and then we can start to look at, so we have 12 of the continuous 7-litre systems. So once they're sort of all at a stable level, the plan is to then start adjusting the feed levels, feed in volumes and rates, and see like where the sort of golden spot is for the HRT.
Brilliant. Thank you. And Chris, I have a couple of questions for you. Firstly, the amounts of paper waste in Yorkshire are lower compared to other regions. Would that have an impact on sourcing and transporting the feedstock?
Yeah, potentially, yes. The graph shows data about facilities receiving the waste materials. So it doesn't necessarily represent where the materials arise. So there could be more waste paper and card in Yorkshire and Humber than shown on the graph. It's just already moving somewhere else to then be processed. So, so that's the first thing. And the second thing to consider is what is the best location for a facility? Is it near where materials are already moving to because the supply chains are established, or is consideration of the life cycle analysis more important? And we don't know the answer yet, and onto that sort of factor. One of the aims that we have is to create sort of a heat map. So where would we want this to locate for hydrogen demand? Where would we want it to locate for feedstock availability, taking account of those factors? So that will determine where it is, and that will determine what the best supply chain is. Hopefully that answers the question.
Yep. No, and another one for you, Chris, that's come through apparently is, do you anticipate much development in hydrogen being used in the internal combustion engine? I don't know if this is you or not. And have H2Boost looked at this option for lower purity of hydrogen?
Yes and yes. We don't know yet how difficult it is for H2Boost, either technically or economically, to reach the 5 nines purity. So for fuel cell applications, so we did during phase 1 actually open the door to discuss with 1 or 2 of the UK companies, as one particular Ulemco, have some co-feed vehicles where they feed fossil fuel blended down with some hydrogen, and their purity requirement, I believe, is around about 97%. So yeah, we can do that, and we will consider that.
Thank you. Another question that's come through is the idea for H2Boost to be retrofitted to existing AD plants, or is it meant as a standalone? Who wants to take that? Was that you, James?
Hi, yeah, I'm very, very happy to do that. So, I mean, this is a trial. This is a scaled trial. So, at the moment, yes, perfectly possible. I think we need to look at the data and the results out of this and see where we're going with it to commercialize it at scale.
Right. And also, what is the composition of household food waste that we're using for the hydrogen production?
So household food waste we used in phase 1. Composition wise, I guess it was very variable. So it was actually from food waste bins. If you mean the food waste blend that we're using at the demo site, so it has paper dust which falls out of the paper process, so it can't actually be used because it's too dusty, it doesn't feed back in. There is some high sugar waste in there, and there is also a lot of drink effluent. So again, quite fairly high sugars, and meat effluent as well, which sounds lovely. But yeah.
Thank you. I've just got a couple more coming in. Sorry, thanks, Sarah. The enzyme that you're including along with the feedstock, is it an enzyme pre-treatment to enhance the hydrogen production?
Yeah, so the enzyme we're using mainly it targets the paper dust, so breaks down the cellulose into sugars so that the microbes can access it and produce hydrogen.
Okay. And then I think let's see the final one. Maybe again a bit more onto you, Chris. Is it viable to just make the biohydrogen, or does it have to have the methane carbon capture to make it financially viable?
In phase one, we looked at this, and it may well be the question is that best use of the digestate, because you then have to move it somewhere, and so there's some sort of life cycle questions that say what's the best thing to do, but that is certainly a configuration we will look at during the second half of the TEA.
Thank you. So I think we'll probably wrap it up there, just to say that we are planning on having another workshop in September, as the demo plant will be up and running from July. We hope to have some initial results by then that we can talk to you. And we're really interested in hearing from any interested parties that are kind of want to hear more about the project or want to work with us all further on this. And there's my contact details if you do want to get in touch. This webinar will be available on the Biorenewables website, and the slides will be available should anyone wish to hear them. And I think one of the questions was the report from phase one that is available online on either the DESNZ H2Bex page or on the BDC website as well, if you want to read about what we did in phase one. A report from phase two will be available early next year once we've completed the project. But for now, I just want to say thank you all for joining us, and we hope to see you again soon. Thanks a lot. Goodbye.